Tonight's theme is COVID-19 research and drug development at SLU. Our speakers are Dr. John Tavis and Amelia Pinto. Amelia her research focuses on arboviruses using animal models to understand disease progression and to evaluate vaccines and treatments with particular interest in the role of cytokines in a protective immune response. She's working with the SLU Center for Vaccine and Treatment Evaluation Unit to understand viral infection pathogenesis and immunity in both healthy and at-risk populations, and is participating in the ongoing remdesivir trial at SLU. So Amelia, please tell us about your COVID-19 research.
Sure. All right, let's see if I can share screens. Got a lot of desktops to choose from here. Let's see. All right. And then let's see if this will work. How'd that work out for her? Does that work? Okay.
Yep, works for me.
So I started here at SLU in 2015. And shortly after starting here, I started to work on a compound screening trial for SARS, the original, which I think is what elevated me to expertise in this field. I have to be honest, I have never given a presentation on data that I have spent so little time on prior to giving this talk. Usually it's many years that I've studied these pathogens, but in this case, here we go. So what I'm going to talk to you today about is first setting up the model of what I view as our traditional way of approaching the research at St. Louis University and how it goes to support the Vaccine Treatment and Evaluation Unit and what we do independent of the VTEUs as well as with the VTEUs. So SLU is one of 9 VTEU sites throughout the country, and they evaluate NIH-sponsored vaccine trials. Sarah George, who is on the phone call, can speak more to the VTU and its background, but ours here at SLU is run by Dan Hoft and his group. And usually what I would say is the general contributing factors for research in this is through assay development for pathogens, diagnostic testing for pathogens, and developing of these preclinical animal models so that we can try out different vaccines and whatnot, which then brings us to the vaccine trials. Now with SARS-CoV-2 and the COVID-19 research, this model that I think that we've been operating on has been completely turned around. It really did start mostly with the VTUs and with the vaccine trials coming, the treatment trials coming here. And from that, we've been working to develop diagnostics, assay developments, and hopefully in the last part of my talk, I'll talk a little bit about the preclinical animal model trials that we have, or the models that we have developing here at SLU. But to say that this is backwards is an understatement. Normally, as John has spoke to, we are much more into the development of these pathogens and understanding the pathogen and pathogenesis and the drugs prior to going into human clinical trials. And in this case, this is completely backwards. So when we talk about the remdesivir trial, and I laugh only because I am as miserable at pronouncing these compounds and these drugs as John is. Not that I have any skill at it, but please bear with me. So the trial, which is a part of the VTU, but is also being run at multiple institutions outside of these VTUs, is the Adapted COVID-19 Treatment Trial, or ACT. This trial is being headed by Sarah George here, who's the PI. It began on February 21st, and it actually closed the first trial on April 19th of this year. And when I say began, I mean began enrolling. So as John spoke to, remdesivir, it's a nucleoside triphosphate analog. And I'm going to give you some basic backgrounds on this drug screen, or on this trial and how it was set up, and then go into a little bit more of how the research here at SLU has supported this trial. So to start with, to be enrolled in the trial, the subjects were screened and found to be quantitative RT-PCR positive prior to enrollment. And in this initial trial, subjects were randomized and either received this high dose remdesivir followed by a maintenance dose for 2 to 10 days, or they were on a placebo. Samples were collected intermittently during hospitalization and post-hospitalization if possible. All non-safety samples that are collected for these trials are sent to DMID, which is where their repository where those samples are stored. And for research purposes and for analysis of these samples, the VTUs are awarded sites or the ability to receive those samples back and analyze them to determine efficacy as well as other research value from these samples. So there is a remdesivir trial going on now. It's called Act 2. It began enrolling on May 5th, and you would have to speak to Sarah as far as the level of enrollment that's currently going on. So this trial had, and here's another one of those fun drugs, the addition of baricitinib, which is from Eli Lilly. It's a JAK1, JAK2 inhibitor with approvals in more than 65 countries for treating rheumatoid arthritis. So this is an anti-inflammatory, and it's supposed to act or thought to act for the inflammatory cascade for COVID-19. So if you think about it as far as remdesivir is targeting the virus or SARS-CoV-2, and the baricitinib is more targeting COVID-19, so the disease aspects of the virus rather than the virus itself. So this new trial is targeting both. In this case, very similar to the first. Subjects had to be RT-PCR positive in order to be enrolled. Everybody, however, gets remdesivir, and only a portion will receive the baricitinib orally, while others will get placebo. So in this study, we've moved past the efficacy of the first drug, and we're looking at the shortness and duration of disease with the second. So how did the researchers at SLU support this and are continuing to support this? And you'll forgive me the analogy, but it was just too easy. But I promise you, I will only speak of this in the acts in this slide. So we have what is the prologue, which is prior to beginning these trials, there needs to be biosafety protocols, IBCs and IRBs. As many of us researchers here at SLU had previously worked on SARS, we did play a decent role in participating in the development of those IBCs and in conversations about the IRBs. We also worked closely to help with the development of and continuing to develop diagnostic assays to confirm SARS-CoV-2 infection. And this third point here about development of protocols for sample collection and useful time points for efficacy and research evaluation, not so much in this trial, as this is much more of a national trial, but normally for the VTU trials that we've been involved in, the research aspects or the research arms of SLU do contribute to the development of these protocols so that we can pick the times where we feel that the viral titers would be the lowest or the immune responses would be the highest. So we contribute to when you would take patient samples and what types of samples to take. In this study, as it was more of a national study, I think that our institution, as well as multiple others, had lots of input that was then, and rightfully so, summarily ignored, as patient evaluation came far before our research interests. So in this intermission that was between Act 1 and Act 2, traditionally, I would say that the research aspects of SLU, we come in and we talk about the protocol revisions, any sort of new tools for diagnostics, reevaluating when samples should be taken. In this case, again, there was very little that research played in the development of the new ACT trials. We really are just here to support any work that's being done. And finally, what I would say is the epilogue, which is yet to be written. Now, I spoke of those samples that are being collected and stored in the repository. So in order for researchers like myself and others to gain access to those, we write proposals and we submit those proposals to obtain those research samples from the trial. Again, these samples are quite valuable, and certainly for efficacy cases, those are going to take priority, but we are actively thinking about and writing these proposals for what we can do with the samples when these trials are done. So, second to that, and in conjunction with the studies going on at the VTU, as well as separately outside of the VTU, how the research at SLU has worked to understand this disease is that we first began looking for what were the initial needs. And as John spoke about this being a new emerging infection, those initial needs really fell to questions of detection for both exposed and infected. So in this case, have you had SARS-CoV-2 before, and do you have an immune response to it? Do you currently have the infection? And again, I will say that this virus is quite different from what we've previously worked on, because what you would normally do is develop the assays to determine if people had been exposed. So the immunological responses, the antibody detection assays, the ELISA assays. And secondary to that, look at the peak viral titers in those currently infected. In this case, we're working again backwards, where the quantitative PCR assays for viral infection and active viral replication were certainly developed first, and the ELISAs were developed after. However, for the purposes of this talk, I'm going to talk a little bit about the ELISAs first. So the ELISA is the assay in which we use to detect whether or not you have antibodies in response to the pathogen. And here at SLU, one of the first questions that people came to us, companies came to us and asked is which viral protein should they target? And this is not actually a very small or insignificant question. So on what I guess is the left of the screen, we're talking about what the virus looks like itself. And the viral protein that we would target most likely would be the spike protein, or this S protein. And so when you look at the spike protein, what it is actually two subunits, with subunit 1 and subunit 2, which also has a receptor binding domain in it. And if you think about developing assays, a high throughput assay that can be rolled out pretty much throughout the world, what you want is to be able to make a protein which has to be folded properly for an antibody to recognize it in a fast, rapid quantity. So what we were asked by multiple different companies was, how much of this spike protein do we need to make? Do we need to make the whole spike protein? Could we make just subunit 1, subunit 2? Which part is important to us for developing these assays? So what happened was we paired ourselves with multiple different companies, both in St. Louis and outside of St. Louis, who provided our research, my research group, and a few other research groups here with milligram quantities of either this subunit 1 protein, subunit 2, or just the receptor binding protein. And I should say that the receptor binding binds to the receptor, the human receptor, human ACE2, and that'll come into play a little bit later. And so what we did was we ran ELISAs. So we coated plates with these different proteins that we were provided and determined from SARS-CoV-2 positive human serum which one of these proteins was best for recognition. To be honest, we provided this data. We did not make the final determinations for these ELISAs. This would be what will be the next generation of ELISAs that are going to be rolled out for detection. The current ones are not as sensitive as these assays are, but they are a bit easier to make the protein than what they provided us here. And for me, as a researcher at SLU, one of the nice secondary aspects of this is now I have what is a great deal of protein from these different parts of the subunit of spike, which I can then develop my own research program on and start to understand what parts of spike are important for the virus and viral entry. So second to that is detection of the virus, and this is detection of SARS-CoV-2. And this is detection of the viral RNA, which is the assay that was the first one to really come out. So way back and I guess this was probably early February when we set up this assay, and I'm showing you probably one of the second or third iterations of this assay. What we set out to do was to determine whether or not the CDC guidelines for how to detect SARS-CoV-2, how to set up that assay and how to run that assay. So that's what this is here, and what I'm showing you here is the real-time PCR data for the detection that was set up and run as the guidelines for the CDC proposed. And what we noticed as researchers was that so what we're looking at here is the red and green line as well as the blue line are the positive controls. And this assay is not very sensitive. And so what we're currently working on now is the next generation of this assay to improve its sensitivity to allow a more sensitive limit of detection for this, so that to avoid false negative detection using this quantitative PCR reaction that the CDC has proposed for screening. And then the next thing I'll talk about is really what I would say my lab and what a few other labs at SLU have been spending a fair amount of their time on, which is infectious virus quantification. Why you'd want this is it is really what is required for understanding antibody neutralization for vaccine development. So when we talk about getting a vaccine for SARS-CoV-2, that vaccine test, one of the first criteria in order to be considered a vaccine is its ability to neutralize virus. And so you have to be able to grow the virus, detect the virus in order to determine whether or not you can neutralize it. You also, I think probably again around February, maybe early March, when people were starting to question about the people who had the infection and were testing positive, how long, if they could detect RNA out 20, 30 days later, were they still infectious? So they were detecting RNA by these qRT-PCR reactions, but it was curious as to whether or not they could come back to work, whether or not they were spreading disease. And so we've been working to develop these infectious virus assays to answer the question as to whether or not, if you can detect the viral RNA, does that mean that you have infectious virus? This assay is also critically important for all the compound screenings for antivirals and therapeutics that everybody is very interested in developing, to be able to find out whether or not in the preclinical animal models or in vitro tests, your compound of interest has an impact on viral replication. You need to be able to detect the virus. This also, and certainly John can speak to this, as well, having an ability to detect infectious virus makes viral research much, much easier. You can start looking at pathogenesis and disease models when you're able to detect infectious virus. The huge caveat to this, however, is that this is the part of research where you have to switch from doing bench research. So all the stuff that I've talked about previously as far as the research at SLU can be done in a BSL-2 laboratory on a bench top. Once we get into detection of infectious virus and growing infectious virus, that's when you switch to your hoods and your PAPR and having to much more restrict access to the pathogen. So there is a huge trade-off to this. It does restrict the number of researchers who are doing this work to a very select few of us. Hopefully we'll have more in the coming weeks, but that is where this is the big turning point between that laboratory work and then detecting infectious virus. So we are able to detect infectious virus. It's kind of a bit hard to see here, but what you're looking at are two 96-well plates. They're plated with cells, and every time you see a little blue spot, that in one of the wells, that is infectious SARS-CoV-2. I do have to say that this was an assay that one of my graduate students had set up to determine cell concentration that was best for detecting virus. And I kind of commandeered it to spell words, which she quite enjoyed. And it does make me quite happy when I see it. But we were developing what were the best diagnostic assay, or what was the best assay for detecting a virus. And it does seem to work, and it works quite well. So now that we're able to and detect this infectious virus, and this was, oh gosh, about a month ago now, again, very rapidly turning this stuff around. We have been able to develop what are neutralization assays. So this is where we're talking about this gold standard of whether or not human immune responses can neutralize or block viral infection. And what I'm showing you is just one assay that was completed a couple weeks ago now with human serum samples from all over the world right now, where we were looking to determine if they were quantitative PCR positive, whether or not they could neutralize virus. And the good news is the vast majority, save one so far, of people who tested positive for SARS-CoV-2 after a period of about 30 days had developed antibodies which can neutralize the virus. So that's a big positive, and it's a big win for us as far as being able to go further and determine what parts of the virus are these neutralizing antibodies detecting, and how can we develop a better vaccine which induces these types of neutralizing antibody responses so that we can look to the people who have not been exposed to develop these responses as well. And so quickly for my last slide, I'm going to talk just a bit about where we are on small animal model development for preclinical evaluation. Really here, we've got two main small animals that we're working with. Hamster models. So hamsters have been shown to be very susceptible to SARS-CoV-2, which makes them a very nice model for studying disease. However, when I put on my immunological hat, this is not an ideal model, as there aren't a lot of reagents available for studying any disease in hamsters. It makes it a bit more challenging to work with. We'd much prefer to work with mice, where we have many more genetic materials available and variations in the mice. There are transgenic mice that express the human ACE2 and whatnot, because mice naturally aren't susceptible. The other way in which we're working to make these mice susceptible is to transfect in mRNA expressing the human ACE2, which we've been able to show does make the mice more susceptible to disease. I should also say that we are collaborating with the Jackson Laboratories, both on the East Coast and the West Coast, who have humanized mouse models to look to see how susceptible those models are to SARS-CoV-2. And we're starting to get some interesting data, but we can certainly use more researchers and more hands on all of that, because it's quite a lot of work, and we're working our way through it. So with that, I will end with, again, what is my favorite focus farming assay plate ever. And along with John, take any questions.
Well, thank you very much, Amelia. So, and thanks again, John. We've got about 5 minutes before we have to vacate the room. So if people could ask any questions. I see there has been some chat back and forth with John about some questions. Oh, and so Ranjit has a question. Go ahead. Oop, sorry. You didn't get unmuted. I'm having trouble unmuting. There you go. Try again, Ranjit.
Can you hear me now?
Yes.
Okay, so I want to just ask a question regarding the asymptomatic cases and the possible latency period this virus might go. And as John has mentioned about the persistence, it's a complete black box. Nobody knows how long it's going to stay, and maybe later on, depending on the immunity as well as aging, it might reactivate. So as you are into this viruses, so what is the probability or possibility that this virus may integrate? And are there any ways, because it's a very complicated thing, so are there any efforts into that direction?
The probability of it integrating is very close to zero. This is an RNA virus. It would have to be spuriously reverse transcribed into DNA in order to be integrated into the viral genome. There are molecular fossils of RNA viruses in many mammalian, including human genomes, but those occur rarely, even on the scale of evolutionary time. So the probability of this causing a direct integrational mutagenesis in a human patient is exceptionally low, almost to zero.
Thank you. Thank you, John.
I think there's a question in the chat.
Oh, yeah, thank you. So it said, this is from Andrew, and it said, what are the false positive and false negative rates for current commercial QRT-PCR tests?
Well, I can say it's pretty hard to know. That there's some reports that put it up at around 40% and others that put it at 10, or below 10. What I can say for the QRT-PCR is, I'm not sure how many people have done that assay, but those CT values, or the point at which you detect replication, is very high, and which means that you're at very close to where levels of contamination are. So it is very challenging to look at that assay and say it's optimized. So it's a little unclear right now as to what the levels of false positive and false negative are, but it is definitely clear that these assays are rolled out for a pandemic and not optimized yet for the level of detection that we would normally develop for something like this.
The only thing I can add to that is the false negatives are also contributed by sampling errors. You really have to stick those swabs up somebody's nose really far. I've heard people joking about scratching the back of their eyeballs. And it's uncomfortable and difficult to do. So there is a false negative rate due to sampling errors in addition to the inherent limitations of the assay that Amelia just mentioned.
So we had two other quick questions. One is just sensitivity of RT-PCR, and are there testing labs in the US using the QRT-PCR based on spike encoding genes?
Not via the CDC approved protocol. The CDC approved protocol requires the nucleoprotein as those primer probe sets that detect the nucleoprotein as the protein that they have to run in order to determine positive versus negative. Now, that's not to say that there aren't other assays. Certainly we have other assays which we've been tweaking to see if we can get a more sensitive test. But right now, for detection, as much as I know, and certainly others could speak to this, but I checked the CDC website last week, and I believe that they are still sticking with the same QRT-PCR assay for detection.
Theoretical level, you can make QRT-PCR tests incredibly sensitive. I believe, although I can't absolutely prove it, that I've had a hepatitis C virus RNA detection system in my lab at one time that was capable of detecting single-digit number of copies of RNA molecules. It's capable of doing that. The current ones aren't there, as Amelia has discussed.
So I think we have time just for one last question before we go. So Yulia has asked if there's a correlation between the number of virus and the ability to infect.
We don't know yet. What we have seen is correlation with the quality of the response and their ability to neutralize virus, but the amount of virus and any aspect that way, we're not there yet. And that has a lot to do with really trying not to interrupt standard of care. I could think of hundreds of things I would like to do, but there is no way I'm going to walk over to a clinician and ask them have these samples right now when in the position that they're in. So we're looking very closely at levels of virus. It certainly seems that there are different levels of virus in different individuals. Whether or not that contributes to spread, we just don't know. It is interesting because there is definitely variation in viral loads in different individuals.
So I think we're out of time. We have to make way for the next talk, next session. But thank you again, both to Amelia and to John. And I think they'd be happy to have you email them with further questions if you want to continue afterwards.
Certainly.
Thank you once again.
Thanks.